What y'all know today? Nothing? Questions? Sure. Well, so we introduced this guy last time, the RAS pathway, and so we want to go on through the rest of these pathways hopefully today, and then we'll have a little time to think things through on Wednesday. So, you know, like I was saying, main point is to know the sort of scheme of the players. We'll meet specific members as we go through these things, but a ligand has to bind to the receptor. Okay, so this is for all receptor tyrosine kinase. There are adapter proteins that are going to modulate interactions with GAPs, and then RAS is activated, and then RAF is activated, and MEK is activated, and then ERKs or MAP kinases are activated. Finally, transcription factors are activated, and transcription is altered. Gene expression is altered, generally resulting in things like cell proliferation, or at least cell gets to stay alive, and also promoting differentiation down different specific tracks. So some of the examples of places during embryology where this receptor pathway is used are shown here. So in 3 different systems, in fruit flies, a lot of the basic players were identified looking at how photoreceptors in the eye develop. In C. elegans, a lot of the differentiation processes for specification of cells were known, or at least the fate that all cells form is known, it's constant. And so they can look at mutations where certain groups of cells don't form or certain organs don't form. So in the formation of the vulva, which is where the eggs are going to be stored for a while after they're fertilized, development showed this. So some of the individual members. So like in this case, one of the ligands, it's called VOS, the receptor is called 7-less, and this adapter protein is called downstream of receptor kinases, and then you have the GEF and the GAT. This is called son of 7-less because that was called 7-less. And then Drosophila RAS, the RAS name was already there, so they just used it. And RAF was already there, so Drosophila RAF and Drosophila MEK, and then various genes of particular transcription factor down here. And so the same thing is true over here, except they have different names for some of them. So different names sort of are which name sort of came first, plus certain organisms sort of have their gene family names already. C. elegans always names genes with a 3-letter name followed by a number. And in mammals, right, these things can set off certain growth factors. They should have said growth factors here, not cytokines per se, but this is true for cytokines as well. Growth factors like epidermal growth factor, fibroblast growth factor set this off. Our adapter protein is called GRAB2, and we have human SOS and RAS and RAF and MEK and ERK and so forth. Okay, so the same basic pathway is used over and over and over again in lots of different systems and lots of different differentiation pathways. The fruit fly pathway was one of the early ones where people worked it out because there was a cool mutation where fruit flies typically will respond to ultraviolet wavelengths of light. They have one photoreceptor, number 7. They have 8 total, but number 7 happens to be the one that specifically can detect UV light for them. And so people were actually training flies to go towards one end of a tube, like a little pipe, where they have UV light, and they would know to go down there because they would get food, like they would drip in some sugar water or something. So all of a sudden they noticed some flies just only randomly go down there. Other flies, after 2 or 3 trips, like they know when the light comes on, food's coming. But other flies are sort of like clueless to it. Well, it turns out when they examined them, they were missing that cell. So that mutation was known as 7-less because the 7th cell was missing. And so then they started looking for other mutations which interacted with it, and it let them work out pretty much the entire pathway. So 7-less turns out to be a receptor tyrosine kinase that's expressed specifically in that R7 cell. So what was its ligand? Well, they found genes which were expressed specifically in or made by the 8 cell, which develops before 7. They're named in terms of their position, not states development just precisely. And so they named the ligand bride of 7-less, boss. And other genes involved, like RUF and things like that, are also receptor tyrosine kinases that respond to that same ligand. But once those 2 things were worked out, they could elucidate the rest of that pathway. And so that's where we have one of the names that has been carried over into all systems, which is son of 7-less, SOS, which is the guanine exchange factor. So that's an important one to remember. So it's downstream, son of 7-less. A lot of these are titled. The very last gene in line is the transcription factor, and it's called 7 in abstention, Senna. So they all are kind of named back to that 7-less. So you'll see that. But SOS or SOS is a guanine exchange factor that name has now been used across lots of species because it was identified in Drosophila first. And then in the C. elegans, I mentioned that there's the vulva, which is derived from 6 precursor cells, and when the eggs are fertilized, they hang out here for a few developmental cell divisions. Well, those 6 cells are triggered by one particular cell, and it sends out a signal of, which is like a growth factor hormone, and it activates LIN12, which is a receptor, a tyrosine kinase receptor, which then triggers these neighboring cells to these 3 cells to differentiate into the vulva. And the ones that don't get the signal just continue to be skin. Interesting mutations here. When these cells don't develop properly, you don't, especially the P6P, which is where the opening will form so the eggs can be laid. If that's not there, the eggs still get in and they start to develop, but they continue their embryonic development inside that structure, basically bursting out of the organ and killing the mother. And that mutation is called bag of worms, because you basically have the hypodermis of the mother worm is all that's left. And then there's a bunch of little squirming larval stages inside of it. Kind of disgusting. All right, so let's go on and look at the highlights of some of the other pathways. The JAK-STAT pathway is one where a lot of interleukins, interferons, those are cytokines involved in, when I say blood cell differentiation, we're really talking here mostly about white blood cells and the immune system. So T cells, B cells, the various other lymphocytes that you have. In the cartilage, FGF receptor 3 is actually activated and sets off a JAK-STAT pathway. So there are some cross reactions between things that would traditionally be thought of as a growth factor and the JAK-STAT pathway. And so here's kind of an overview of that. You have your ligand, again, causing dimerization of receptors. The distinction here is that the kinase is not the receptor. It's associated with the receptor. So there's a specific linkages between the receptors and various members. So there's several of these JAKs, and they will bind to a JAK-associated receptor, or the receptors are typically going to be named for the ligand, which would be, like in this case, prolactin receptor. And that brings about dimerization, and the kinase, the JAK part, will then phosphorylate itself, its counterpart, and the receptor part, and phosphorylate STATs, which are signal transducers and activators of transcription. And there are a number of these different proteins, which will dimerize and enter the nucleus once they've been phosphorylated. And so they're phosphorylated on tyrosine. So the JAK is a tyrosine kinase, but it is not a receptor tyrosine kinase. But it's associated with a receptor that is. Okay? Or it is associated with the receptor. Anyway, end story is it always activates transcription. All right, so in receptor tyrosine kinase pathways, what are some things that can go on in development that they control? We've looked at a few already. Eye development in fruit flies, self-differentiation in C. elegans. What types of abnormalities or what types of things can go wrong with these pathways? And so you should think about what would happen if the pathway was activated in the absence of its ligand. Okay, if the receptor was simply on or could dimerize without the ligand, it would essentially be running rampant, and that's a constitutively active situation. So you can have receptors that are essentially activating the signal cascade without any signal. You can also have constitutive active members of any part of that signal transduction cascade. So RAS, the member of that pathway that we typically call this the RAS pathway, we said last time RAS was the first oncogene to be identified because it's mutated, and when it's mutated, cells will proliferate and become tumors and can form cancers. Well, why does RAS do that? How does it make the cells proliferate? Well, all of those mutations are actually constitutively active versions of RAS that can bind to GTP without needing a gap. So they don't need the upstream signaling, they just come on. Then they activate everything that's downstream of it. Okay? So you can have constitutively active members of the transduction pathway. You can have dominant negative or dominant inhibitory mutations. This would block signaling. So this has the ability to bind to the ligand, but the inability to transduce the signal. And that can happen to other members like GEFs or GAFs. If they're not activated properly, they are not going to respond to the signal properly. They won't proliferate the signal, propagate the signal. So you can think about mutations which could stimulate inappropriately or fail to stimulate the pathway. Okay? And here are some examples of FGF receptor. This is a constitutive active mutation which causes the cartilage to differentiate prior to proliferating enough, and you get faulty cartilage deposition, therefore faulty bone formation. So the limbs are shortened. So some of these are the cause, some FGF mutations, both in the ligand and in the receptor, are the cause of certain types of dwarfisms. All right, so the SMAD pathway. Let's look at the SMAD pathway. Now, this is a different category, all right? So when you're putting things in big pictures, RTKs, that's what we were talking about there, okay? Receptor tyrosine kinases, RAS, MET, MAP, okay? Now we're going to talk about serine threonine kinases and the family of ligands that activate them and their signal transducer. And it's the transducer that gives the pathway its name, SMAD. SMAD is one of the middle proteins, like RAS. It's one of the transducers, okay? So these things are involved in a lot of embryological processes, like the differentiation of mesoderm, the specification of ectoderm, and from neural ectoderm, right, to make it be skin versus neural, and dorsal specification, which has to do with the specification of cells that are going to be on the dorsal side of fruit flies, which also is happening on the ventral side of mammals. So there are three major, as this slide might allude to, three major groups of these ligands. There's the TGF-beta families, the BMP families, and the activin families. Well, they're throwing activin in here. So you have alignments here. This is a one way of representing the family groups or the closest relationships. So the BMP sort of sub-superfamily, TGF-beta is the superfamily. The BMP membership is pretty large. There's a lot of more closely related to them. Activin is down here, is generally kind of thought of as its own little group, and then the TGFs. And then there's some that are sort of like, we don't really know where they go. The data's not in yet, or at least it wasn't in when they made that picture. So you can roughly think of it as, you can subdivide it into TGFs and BMPs, and then the TGFs can be divided into TGF and activin. And the BMPs have a lot of different names. BMP is in it. BMP means bone morphogenic protein. They do a lot more than bone, okay? You can also see that there's just like lots of them. 7, 8A, 8B, 6, 5, 12, 13. They have lots of different names, numbers go along with them. Okay, so why is it the SMAD pathway if the ligands are BMPs or TGF-betas? Why wouldn't it be called the TGF-beta BMP pathway? Well, in fruit flies and in coeliacs, researchers looking at various mutations. So DPP, let's learn a name. Decapentaplegic. Deca means 10. Penta, decapenta, 15. Plegic, not working. 15 malformations. Drosophila has 15 segments. Each segment's identity is messed up in mutations in DPP, and that's how it got its name. And we'll never say decapentaplegic again. It is a ligand, and receptors for that, these are the names given because of the mutant phenotypes. Remember, fruit flies like to name it after the phenotypes. So there were vein malformations in the wing. I don't have any idea how they gave it the name saxophone, except that when a fruit fly larva is developing, the cells in the body sort of curves and then comes back. And I think in this mutation, it doesn't come back. So I think like a saxophone comes and curves up. It's kind of like how they got the name. And punt. I have no idea. They just punted on that. Right? They knew that these things, decapentaplegic, was activated early during embryonic development and it was responding to signals that were placed into the embryo or the developing egg by the mother. So there were maternal genes, proteins and RNAs that were being placed into the egg that must be interacting in this pathway. So they looked for mutations that would block mutations in decapentaplegic. So if this is messed up, could you find a mutation downstream of it that could be activated without the need for DPP, right? Basically, could you find a constitutively active version of one of the genes downstream that would compensate for missing the inactivity up here? So if DPP wasn't really working well, could you have a gene downstream which was working a little too well and compensate? So they called those genes mothers, and this would need to be a maternal gene, mothers against decapentaplegic. MAD, as in mothers against drunk driving, the MAD thing, they use that little idea, so MAD. So MAD was identified as a mutation and then eventually the gene. Along around the same time, the C. elegans people appear. There is a stage of development that the worm can enter into if it's starving. So if there's not very many bacteria in its environment, that's what it eats, bacteria, it can essentially pause its embryonic development so it stays in an immature form and it kind of goes into a state of suspended animation called a dower larva. Well, there are mutations in the formation of that. Sometimes it would happen even if they weren't in, they weren't starving. They would just form dower larvae. So those worms are smaller than usual. So a bunch of the mutations were called small because the larvae were small, dower larvae. Okay, and everybody thought they were working on their own thing until they started sequencing the genes and realized that the protein MAD and the proteins of small were basically the same protein, same amino acid sequence almost. So they combined it to create SMAD. Okay, mothers against decapentaplegic, MAD, small, SMAD, right? Everybody gets their complete name in the name, which is unusual. Well, that's great. So then they started finding more and more and more stuff going on. It turns out that these SMADs don't just do one thing. There are 3 flavors of them. There are regulatory SMADs, co-SMADs, and inhibitor SMADs. So you've got R-SMADs, co-SMADs, and I-SMADs. All right? And so in Drosophila, they came up with, okay, so the co-SMAD, Medea, that's some Greek mythological thing. I'm not sure what that means or what she did or he did or whatever it was, mythological. And then the inhibitor, daughters against decapentaplegic, not dads, but it's daughters against decapentaplegic. I call it dad. All right, so cool names. Maybe that helps you remember where they are. But what are they controlling? Lots of different things in development. So how does the SMAD pathway mechanistically work? You have 2 receptors, which are different. And that's why you have like thick vein and saxophone. There's actually two different receptors. The ligand brings them together. So it brings together 2 type 1s and 2 type 2s. Okay, so they're called receptors, they're called with a Roman numeral 1 and Roman numeral 2 after the name of it, or whatever it is. So like TGF beta R1, TGF beta R2 would be the name. Okay. So you have those 2 receptors. The type 1 binds to the ligand first, then that recruits the type 2s. The type 2s are the enzymatically active members. When they get into the complex, then they go about phosphorylating each other as well as the type 1s. So everybody gets all phosphorylated, and the phosphorylations are going on serine and threonine amino acids this time. So these are called S, T, K, S, T, keep the slash in between there, serine, threonine, kinases, okay? And then what do they do in turn? They cause phosphorylation of the various SMADs. They're going to phosphorylate the R SMAD. It's going to be on the next slide. I'll show a little bit more in depth. There we go. They're going to phosphorylate the R SMAD. Phosphorylation of the R SMAD actually kicks off the inhibitor SMAD, not shown because they didn't know about it yet, and allows binding of the co-SMAD, and then they can go into the nucleus. So the co-SMAD has the, together they have a nuclear localization signal, so that lets them get in the nucleus. Once in the nucleus, they're transcription factors. So it's the co-SMAD plus the SMAD, the R SMAD, are transcription factor. Okay, so you have R SMADs and I SMADs are together. Then phosphorylation comes about. So when it's phosphorylated, now the I SMAD leaves and a co-SMAD joins. Okay, then it can go to the nucleus. The co-SMAD is always 4. The co-SMAD is 4. The R SMADs are different depending on the 2 subfamilies. So the TGF-beta subsuperfamily activates SMADs 2, 3, and BMP subfamily either does 1, 5, or 8. Okay, not 2 and 3, 2 or 3, not 1 and 5 and 8, 1 or 5 or 8. Okay, so they're just different members, and this is how you get different effects. They all bind to 4, and they all go to the nucleus. So, receptor, heterodimeric, heterotetrameric, really. Type 1s bind, type 2s phosphorylate. Who do they phosphorylate? Everything. Each other, ones, and then they phosphorylate the SMADs. Bottom line, SMADs get to go to the nucleus. Turn on some genes. Okay. Again, the I SMADs have names, numbers really, 6 and 7. And so 6 binds 158, 7 binds 2, 3. In the unphosphorylated would be another way of writing hypo. They like to write hypo because it just means less phosphorylated. They're not sure it's completely unphosphorylated, then we go there. And then once they're phosphorylated, SMAD4 gets to bind. So how do you regulate some of these pathways? Okay, so there are a couple of different ways. One is you could bind to the ligand and prevent the ligand from binding to the receptor. Okay, you can do this with anything, but this has been shown to be important because these other genes, noggin, cordon, follistatin-like gene something, and short gastrulation and twisted gastrulation, these are mutations that affect development. And then people figured out, well, how do they affect development? Well, it turns out they have to bind to these ligands. So you don't have that with the FGFs and the EGFs and stuff like that. This seems to be a way of regulating the SMAD pathway. So noggin and cordon bind to GMPs, follistatins and friends bind to activins. Short gastrulation and twisted gastrulation can bind to decadent collegiates. This is the fruit fly genes, frog genes, human genes, mouse genes, frog, human, mouse genes. So that's one of the major ways of regulating this pathway is to eliminate interaction with the ligand. So we'll meet some of these players as we go into frog development, but bottom line is they're all working the same way. They're binding to the ligands, preventing the ligand from activating the receptor, which then prevents the whole cascade. All right, so any questions on some ads? Let's move to another family. Hedgehog, because hedgehog. So why is it called hedgehog? So here's a wild type fruit fly larva, and here's a mutant hedgehog mutant. And you can see it's much smaller, and you can also see that the little stripes that are there are actually still the same width. They're just not all there. But the entire width of the stripe has little denticles on it, little protrusions, and only the back half is supposed to have. So they got extra little bristles, right? Like a little hedgehog. So somebody named it hedgehog. And then at the same time, there was, or just around that same time, was the Sega games with the Sonic the Hedgehog. Okay? So once this gene was found in fruit flies, by this time, scientists realized that if you find a gene in a fruit fly, immediately look for that thing in every other organism you can find and get, right? Clone it in every little thing, right? Like in the song, if you listen to the song yet, clone the gene and everything, right? So homology screens out the wazoo. Everybody's looking for the same gene in every organism, and that's where they found hedgehog. So hedgehog is the first one in fruit flies, and then they found it in mouse, so they called it Sonic hedgehog. And then in mouse, it turns out that there's 3 different genes, so they named the first one Sonic hedgehog, and then they had to go with like actual species of hedgehog, so they went with Indian and desert hedgehog, which I guess are 2 species of these things. Anyway, I didn't know there were that many different hedgehogs. They are adorable looking. So some of the places that they play, the majority of their roles are in limb development and polarity, as well as in development of the nervous system. So look at your hand, the one you're not writing. You can assume anatomical position, right? Which is you would be on your back, but palms up. So on your hand, you're going to have your thumb on one side, pinky on the other side. So this is anterior thumb, and pinky is posterior. Because if you were to rotate your hand this way, which is how you would normally have it, the thumb is pointing forward. And in humans, pointing forward is anterior. And only in humans. In every other organism, this is anterior towards the mouth. And think about your dog or your cat. Do they walk around with their mouth pointing down? No, they walk around with their mouth pointing straight out, right? That's anterior. So this is anterior for us too. But then they kept doing it. Instead of calling this ventral, they keep calling it anterior. So for the medical school folks, just realize that you're going to enter a human-centric world where ventral for everyone else in the world becomes anterior for you. But that polarity is hedgehog. Okay, put hedgehog in the wrong spot, and you can get 2 thumbs or 2 pinkies, depends on where you put it. It actually controls the posterior side, so you get 2 pinkies if you put it anterior. The proteins, just to show you, they are really different in shape. They do share a structure where they have these 3 things right here, these little ribbons that I'm kind of pointing on. They're called beta sheets. And that's where they make their interface with the receptors. So that's where they share some commonality. Now, the hedgehog was really hard to work out, the pathway. And part of that was because it's one of these signaling molecules that is very limited in its range to only maybe 1 cell diameter or 2, or very few anyway. And that's because when it's secreted in a coat of heparin sulfate proteoglycans. That's the little fish hook looking things. And so it is also has a lipid tail and a cholesterol. It has palmitic acid on one end and cholesterol on its other end. Also, the protein has to be proteolytically processed. So when people started making this stuff in a test tube and trying to inject it into different critters, it's not processed right. So if you take the fruit fly hedgehog and put it in frogs, it's not processed right. You know, they're both eukaryotes, but the machinery, the enzyme doesn't recognize the fruit fly version of the protein and cut it. So it doesn't get processed. If it doesn't get processed, it doesn't get alkylated, and it doesn't get shipped out right. So it's kind of a weird thing. So as it gets expressed in these exovesicles, so to speak, or into these lipoprotein assemblies, that's going to be important for its function. If it's not secreted like that, it won't bind its receptors. Okay, so where in the embryo does it work? Limb buds, nervous system, that's a limb bud, that's a limb bud down there. Actually, this whole thing is the limb bud, this little mound. And it's only in the posterior tip of it, or side of it. A lot of weird things happen if you mess with hedgehog. These kinds of chemicals were known for a long time to cause birth defects, especially not in people, because people don't go out and graze, but defects in grazing herbivores like sheep and cattle. And so this one, Veratum californicum, this thing right here, produces two different inhibitors of cholesterol biosynthesis. And if you're not making cholesterol right, you won't necessarily modify the hedgehog right. And so gervine and cyclopamine cause things like this. Cyclopamine, cycloptic. So hedgehog, one of its major things is to make two sides to the nervous system instead of not. So signaling early on prevents that from happening, or lack of signaling early on prevents that from happening, and you don't get two optic vesicles, you just get one. So one eye. I don't know about the foot. That's a hoof. Maybe that's an ear. I'm not really sure. Poor guy's messed up. This would happen to a human fetus also if you ingested those compounds, right? So, don't do that. So working out the pathway of hedgehog was a pretty long process because it doesn't bind to a single protein for a receptor. It's secreted in a weird way. It doesn't diffuse very far. So it was hard to recapitulate its processes in tissue cultures or in xenophys culture or anything like that. So it was largely worked out through genetic dissection in fruit flies. So all the genes are pretty much still have the fruit fly name. So the receptor for it is patch. Okay? Whatever patch does, and there's accessory proteins here, but whatever patch does, it prevents the inhibition, or what it would normally do would be inhibit smoothing. What does smoothing do? Smoothing gets degraded. Okay, what would happen if smoothing didn't get degraded? If smoothing does not get degraded, smoothing activates and becomes a phosphorylated protein. Gets phosphorylated by just a regular old kinase called PKA, but then it acts to block the signaling here of all of these proteins, which are just proteins hanging out on the cytoskeleton, the microtubule cytoskeleton. Okay? If it blocks their interactions, one of the members, called CI, goes to the nucleus and acts as an activator. If smoothing gets degraded, none of this happens. This thing gets phosphorylated instead of smoothing, and it gets cut when it gets phosphorylated, and half of it goes to the nucleus and acts as a repressor. Okay? It already sounds like it might be complicated, right? Not really, but might be, right? I'm joking. It sounds freaking ridiculous, right? So you can imagine, just put yourself in the guy's shoes, the gal's shoes, and the graduate student's shoes, and all the people who were trying to figure this shit out. They were like scratching their hair and like, what the heck is going on? It can't be an activator and a repressor. And they have all these weird names, which we'll look at later. But It's one of the first times where a new paradigm came into play. You basically have an activator of an inhibitor of an inhibitor. So without the activator of the inhibitor, you inhibit the inhibitor. When you activate the inhibitor of the inhibitor, now the inhibitor can go ahead and inhibit, which in turn allows the activation of something else. Okay? It's like a whole bunch of on-off switches. And this was the first time where basically this gene's function is to inhibit. And if you inhibit, so it's inhibiting, right? If you bind hedgehog to it, it doesn't inhibit anymore. So you've inhibited an inhibitor. And now you have activation of this, but this is actually an inhibitor of phosphorylation. And when phosphorylation is inhibited, now you can get activation. Okay? So it set a new paradigm of thought, which as you can you're confused already, right? It's like the scene in The Princess Bride. You guys do know that movie, right? The guy's like, clearly I can't choose the one in front of you, and I clearly can't choose the one in front of me, so therefore blah, 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 right? It's that kind of logic. And so it's like an inhibitor, back and forth. It's crazy, okay? So just think about pathways like hedgehog are inhibiting inhibitors of inhibitors. Same thing happens with the mouse version, Sonic Hedgehog, except we changed a few of the names, but not too many of the names. So the only one that's really changed much is the last one, Glee instead of CI. Same protein. Interchangeable. All right. We got 5 more minutes? Yeah. One more pathway then for today. The Wnt beta-catenin pathway. Oh, it's crazy, right? How can we do this many pathways? And I'm not asking you right now to remember all the things that they might be doing. I want you to remember the names of these pathways. Okay? So Wnt is a hybrid of two names, the fruit fly wingless gene and the mammalian gene called INT, which stands for integration site. It's where a viral integration happened that caused a lot of tumor formations, specifically breast tumor formations due to viruses in mice, not in humans. And the other player in the name, beta-catenin. Beta-catenin is usually found or usually thought of as its role of connecting cadherin to the actin cytoskeleton. Okay? But in fruit flies, it was first identified as a mutation, gave a phenotype they named armadillo because the segments aren't forming right. Instead of 15, you get 9, right? 9-banded armadillo. So you have some overlap in the various names. Eventually, it was decided to just combine these things to Wnt, and beta-catenin kind of won out in the naming scheme for that. We find that they are one of the first signaling pathways used in the specification of dorsal in frogs and in mammals and other vertebrates. And in fruit flies, they're involved in establishing the segments, the 15 segments, specifically the polarity of that, the head end versus the tail end of each segment. So that's segment polarity. So they're acting over, kind of like hedgehog, they're acting over short distances. Here you go. If you want something to make you go to sleep at night, just read that. And if you want to make someone smack you upside the head, just keep reading it to them over and over again. But Wnt can do some cool stuff. I misspelled stuff. So one of the early experiments in Xenopus, or one of anything, when a gene was found, the Xenopus people can inject it into an egg. So everything just got injected into an egg. And it would always get injected into the egg and see what would happen. And in the case of activating the Wnt pathway, you can make the embryo have 2 heads, actually 2 partial spinal cords and everything. So this is an early neurula stage, and you can see there are 2 neural tubes forming. And here it is later, where you get 2 heads. In fact, this is a fairly complete one. Most of them don't have 4 eyes. They just have 3 eyes. The middle one kind of merges. But that's pretty freaking cool, right? 2-headed frogs. If the frogs weren't scary enough. So how does the Wnt pathway work? Who are its players? Its receptors were originally found in fruit flies named frizzled because the bristles patterns are messed up. One of the signal transducers also had messed up bristle patterns called disheveled. Ears messed up, right? The fruit fly version of this gene goes by the name of ZEST-white3, ZW3. But it wasn't known what it did until its complementary gene or its cognate gene was found in frogs. Glycogen synthase kinase. This is a enzyme that normally just functions in like sugar metabolism. Glycogen synthase kinase regulates an enzyme called glycogen synthase phosphorylates. Well, it phosphorylates a lot of things, as it turns out. One of the things it can phosphorylate is the protein beta-catenin. So the way this pathway works is in the absence of Wnt, the receptors are just hanging out in the membrane, and beta-catenin is being produced, and it's being either interacted with the cytoskeleton, which they're not showing you. So if it interacts with the cytoskeleton and cadherin, fine. But if it doesn't interact with the cytoskeleton, it forms a complex with several other proteins and it becomes phosphorylated, and then it gets targeted for degradation. So there is never any excess beta-catenin hanging out in the cell. You have active exactly as much as you need for the cytoskeleton. When Wnt binds to its receptors, they interact with disheveled, and it's not clear still how disheveled does what it does, but one thought is it basically recruits disheveled to the receptor, and disheveled in turn recruits GSK3 and the auxin complex to the receptors. Therefore, beta-catenin is left alone, and beta-catenin may accumulate. And if beta-catenin accumulates in excess of what it needs for cell adhesion, it goes to the nucleus. And when it goes to the nucleus, it interacts with the transcription factor and causes that transcription factor to activate gene expression. Normally, that transcription factor is there, but it's not activating. So it's a co-activator of transcription. Okay? This is called the canonical Wnt pathway. It's the only one I want you to remember. Okay? This is just fun, but who gives a crap? The canonical Wnt beta-catenin pathway. Again, you have a inhibition of an intermediate. You have disheveled, which would normally inhibit or could inhibit GSK3, which is inhibiting beta-catenin. When you activate the receptor, you inactivate disheveled's inactivation, cause it to inactivate GSK3, therefore beta-catenin survives. With Wnt, beta-catenin alive. Without Wnt, beta-catenin dead. Okay, think of it like that. So that's why it gets in the pathway. Wnt, beta-catenin. With Wnt, you get beta-catenin. Without Wnt, you don't. Okay? When you have it, it's a transcription factor. This is the first thing that is seen to differentiate cells on the dorsal side versus the ventral side in frogs, for example. The beta-catenin protein begins to accumulate in the nuclei on the dorsal side. The only weird thing is there's no Wnt protein on the dorsal side. Okay? So beta-catenin is something else is somehow activating this pathway. Well, it turns out that there's another modulator of disheveled, but anyway, that's just a complication of the story. Wnt is like hedgehog. It's secreted, it has to go through a weird process, and you can prevent it from interacting with the receptor in a lot of ways. So these are several members of the inhibitory sort of side of things. These all bind to Wnt, okay? This looks like the receptor, but it isn't. This is specifically just binds. This just binds. This just binds. They just bind to it and prevent it from binding its receptor. Okay? So that's how you inhibit that one. You bind to it, block it. Okay? Bind to the ligand. All right, so we'll stop there. And Wednesday, we've got like a couple of things to mention, but we'll have some time for questions. So maybe on Wednesday, if I go, hey, any questions, there'll be a peep or two out of just a peep or two. Okay?